It's a pleasure to have Gretchen Hofmann here today from one of our sister campuses slightly to the north, Santa Barbara, where she is now the chair of her department. And over the years, as we've been hearing more and more about increased CO2 emissions and the problems that poses in terms of atmospheric temperature and then now ocean temperature, the other consequence of that is that more of that CO2 is dissolving into ocean waters, making it more acidic and creating increasing challenges for the marine organisms and ecosystems that live there. So this is what Gretchen spends most of her time thinking about nowadays, and it's a pleasure to have her here.
Thanks, Paul. Thanks, everyone. It's really a pleasure to be here. I've never been to UCLA as a speaker before, so this is great.
Wow.
Thank you so much. And thanks to Grace and everyone for getting me here. It's really a pleasure to come and share some of the work we've been doing in marine systems. So this is going to run off a stick, so I hope we don't have anything weird happen. Let's see what happens here. So, as Paul said, I've been working sort of in the area of ocean acidification for a little while, and what I want to talk to you about today is kind of really where that journey took me eventually. These days, for the students in here, people ask me, well, what are you? What do you do? And I don't really have an answer anymore. I think I started out sort of being an ecological physiologist, and now things are so multidisciplinary that we're working with oceanographers, with sensors, and reaching out a lot of times to molecular biology. And so I've started to think about telling people that I'm a global change biologist, and that sort of is a very helpful descriptor for me, because what we end up doing is while we are always thinking about organismal function, we tend to be thinking about environmental change. So what's the abiotic environment doing? And for us, it's like the physico-chemical environment for benthic marine organisms is what I'm going to focus on, and a bit about the plankton. And then we go on from there, and we know that, yes, we've learned over time using sensors, and we've known this, but we really know it now, there's a lot of environmental heterogeneity. So depending upon where you are in the ocean, there's a lot of temperature and pH, carbonate chemistry change. Not everything is as smooth as the open ocean. And we also then take some of that data, and we've been using it to parameterize our experiments. So now, instead of setting temperature and PCO2 to what we think the atmosphere is going to be 100 years from now, we can actually change laboratory conditions to match exposures, and we can conduct much more informed experiments that then might layer on the amount of change that we would see under a global change scenario. And while we do these experiments, we ask things about biological responses, so lots of different kinds of response variables. And what we're really trying to do, hence all the little question marks, is when people ask you, what do you think is going to happen? A lot of the work we're doing right now is really thinking about ecologically important species, and in some cases economically important species or fishery species, where we wonder, what's going to happen to the red urchin fishery in Santa Barbara, where we have a very healthy urchin diving fishery up there, to get uni, the well-known sushi, and also now apparently in the last several years, like the flavoring of fishes in restaurants, so you'll order something of a beautiful fish that's seasoned with uni reduction and blah, blah, blah. So uni's become much more valuable as a fishery, so we're kind of always working across that space. And ultimately, we kind of bring it all back together and try to talk about the biology of the organism in its environment on a trajectory of change, but knowing that its function right now is somehow grounded in environmental exposure of today's organisms, present day conditions, and that organism's environmental and evolutionary history. So it tends to get to be all one big ball of thinking about how an organism is interacting with its environment. And in this arena of global change biology, I'm obviously working in marine systems, but as you all know, this is a pretty big area in biological sciences right now. And I teach a class in global change biology, and this is like one of the things that we start talking about, that if you go into the literature, you can see 4 dominant responses to environmental change, with geographic range shifts probably being the most dominant signature. acclimatization, we call it, using physiological plasticity to try to get used to what changes are happening and what could come in the future. True adaptation, and then obviously extinction, which is not a very good outcome here, but it is one of the responses. And I kind of got really interested in thinking about these responses, especially plasticity and adaptation and how they map onto the populations that I was studying anyway. Like, we were working on everything from Antarctic fish to sea urchins, and it became clear that, you know, the way that these responses at the organismal level would play out could be individual population, multiple populations, or species range phenomena. And across those, you could look at physiological plasticity in individuals, or you could look at local adaptations of populations up and down the coast. So, you know, seascape genomics, Steve Palumbi's group at Stanford has been kind of pioneering that area. It's sort of like landscape genomics or landscape biology, asking, do oak trees, Victoria Sork, do oak trees have different structure? Well, do marine organisms have different structure that lends them some adaptational capacity to environmental change? So I got really interested in thinking about, do marine populations have these plastic responses, and is any of it hardwired? And so today I'm going to talk to you about one experiment that a new body of research we've been working on, and it's the first time I've talked about this data, so it'll probably end in tears, you know. But it's kind of it leaps off of work that was done by Morgan Kelly, who's now a professor at Louisiana State. And Morgan was a population biologist who trained up at UC Davis, and she called me up one day and said, you know, I really want to think about population biology in a climate change context. And I said, ooh, I've got a winner for you, you know, because we can go out and get purple sea urchins from different adult populations up and down the coast. We can raise their progeny in the lab and query them really easily. And we have all these genomic resources, because purple urchins, Strongylocentrotus purpuratus, was one of the first marine calcifying benthic invertebrates to have genomic resources. So things all kind of lined up, and basically we did some really neat studies that showed us that, firstly, there's a lot of standing genetic variation already in the urchin populations that lends their progeny to having some genotypes that can handle future gnarly conditions, warm temperature, low pH, altered carbonate chemistry. So that alone was a neat observation, because up until that point I was giving lectures of, well, everything's going to die, and thank you everyone. There are cookies and coffee for everyone. So it was really bad. And when we did, so this came out in Global Change Biology 3 years ago, and it was really a moment where we were with others asking population level questions, querying organisms, what's going on at the population level. And it was a really nice turn. And as a result of those experiments that Morgan did, we also had two observations that were really old-fashioned. This is what I love about this. Like, we're going old school on this. There is nothing new here, people. This is invertebrate zoology that's been done for hundreds of years, really. So first we saw strong evidence of maternal effects, and we went, well, of course, because invertebrate zoologists at Freddie Harbor, led by people like Dick Strathman, have been measuring egg size and provisioning for a really long time. But we saw a really interesting signature of how the female coming from a different location was performing, her progeny were performing. And we also saw that there was really strong evidence that the sires, depending upon where they came from, also contributed some tolerance to the progeny. So all of a sudden we had evidence that sperm mattered. And, you know, really, unlike eggs, a lot of times people think about sperm as just like, well, whatever, you know, packages of DNA that swim and boom, it's over. As soon as the acrosome is over, you just think the sperm is just, well, delivering, you know, the other part of that gets you to 2N, right? So, but we saw a signature that said that it really matters, that the sperm was somehow different. There's differential performance there in terms of how the progeny turned out. And so now we've got like a two-pronged project to ask some very simple questions that we now call who's your mommy and who's your daddy, because the different exposures of the parents change the performance of the progeny in a climate change context. And we're kind of using some very worn techniques to look at this, but we're also kind of going in some new directions to think about mechanism. So today I'm going to show you some of the newer experiments we've been doing on purple urchins. And really we're digging into mechanisms that can confer physiological plasticity, nothing fancy here, but in the context of existing natural variation in the pH and temperature environment that these animals are from, and then also what might happen in the future. So I'm going to present to you one experiment that we did. It's going to be about conditioning females and males, raising reciprocal cultures to test the progeny. Good old-fashioned larval biology is fully at the table, and we're also going to talk about some response variables, but the transcriptome for us as a trait is one thing that we're looking at very carefully. And at our peril, we are starting to ask questions about how the transcriptome could be adjusted and through some epigenetic mechanisms. And today I'm just going to talk a little bit about DNA methylation and whether or not that could help us understand what's going on here mechanistically. So that's the new part, and we'll talk about that when we get there. So, I mean, everybody's pretty familiar with transgenerational plasticity, but probably, I don't know, like in the last 5 years in the literature, you can go find people working on everything from, I think there's even a study on seagrass that just came out, but fish, invertebrates, looking at how conditioning of the parents confers different tolerances to the progeny. So these are two studies on copepods and a lolligos squid, where depending on what the parents experienced, they produce progeny. The babies have different abilities to tolerate abiotic stress. And this is kind of not a huge surprise because we've been studying a lot of this in invertebrate zoology with regard to temperature, et cetera, and provisioning of eggs. But these studies have been looking at temperature combined with low pH or combined with hypoxia. So actually trying to frame the transgenerational environment in a way that matches the natural environment really well, and then with a trajectory to the future. So this is kind of global change biology, transgenerational plasticity, if you will. And so one really familiar way that we've always thought about this is maternal provisioning. So these are some data from a really nice review from Amy Moran, where she's just describing here for groups of different urchins kind of what's in the egg. So protein, lipid, and carbohydrate shown there in the different colors. And this is really classical, that we think that in invertebrate eggs, the size of the egg matters, the amount of protein and lipid in there seems to influence the performance of the future progeny. So this is classic, going on for decades biology. But we've never really thought about it in a global change context, in a natural environment setting. So maybe less familiar to us is kind of an emerging field right now that people are calling ecological or environmental epigenetics, or epigenomics, if you're looking at the whole genome. And by way of backstory, this is really an assessment of mechanisms that can modify gene expression. And in the work that we're doing, we're thinking about how it might do this in marine metazoans, but in an environmentally induced way. And really, one of the mechanisms we're going to talk about today, there are many of them, modifications of how the genome can be changed using small RNAs and other ways, but methylation seems to be a really strong one. But we want to know if that's responding environmentally. And the only reason we can do this, honestly, is that in the last 5 years or so, genomics and epigenomics have become really central to human biology and human medicine. So 5 years ago, I could never presume to do a little test that I'm going to show you, because companies have come to market with kits that are really easy to ask. So, what's the global level of methylation in these cancer cells or these non-cancer cells? And so all of a sudden we started seeing, there's one company called Gentech that produces kits like crazy. So for $500 you could do something that you could never have done years ago. So a lot of this we're just pretty much slurping off of human medicine right now and using these approaches in our laboratory. And I have to say, grad students who are ever thinking about this stuff is super easy. All you need is a spec and a plate reader. And it's not the answer, it's not like a silver bullet kind of thing, but it gives you another level of inquiry that's kind of interesting for us in this context. So the only reason we can talk about this right now is because tools are coming to us from other fields. So just a little tiny bit of molecular biology chemistry. So DNA methylation is where an enzyme, a methyltransferase, sticks a methyl group onto one of the bases in DNA and basically converts things from a thymidine to a uracil. And in the systems that have really studied this, what they found is that in a regulator zone, high degrees of methylation suppress gene expression. So the idea is that in cancer biology, for example, if there's a methylation site, it alters downstream expression of the exons in a way that drives cancer, for example. And but one of the interesting things about these signatures that are called epigenetic marks is they're not permanent. So they can possibly change over the lifetime of that individual or that cell type. And so there's a lot of talk about how epigenetics could influence human health over the arc of your lifetime. So, you know, my constant drip exposure of 1 cup of coffee every morning, come hell or high water, is somehow changing my genome through driving those methylation patterns. If I were to stop that, take the caffeine out of my body, it would probably change my genome again by changing those epigenetic marks. So a lot of this work is also being done in ecotoxicology fields to try to figure out how environment acts with the genome. Hi, how are you? So really, this field is kind of opening up. And I think the place where environmental epigenetics is really coming on is in ecotox, really. But for us now, we're going to kind of think about it in more of an ecological context. And in our case, specifically in this project, we're asking, can these methylation signatures alter the way the genome is expressed in a way that's environmentally useful or relevant? So, in order to teach myself some things, I wrote a mini review, and it was really fun because it was a really new field, and there were only like 10 papers I had to read, so that was kind of fun. It's the first time ever I've written anything where I could put everything I needed to into one table, so that felt kind of great. I postdoc with George Somro when I was much younger, and one time I was talking to George about taking orals, and he said to me, yeah, you know, when I took my orals exam, I just had to read a couple books, and that was everything you needed to know. And I was like, that's not fair, because the literature just gets more and more complicated. But this was kind of like my George experience. I finally had it, where here was just a small area that was emerging. So people have been looking at DNA methylation in marine metazoans, and we're finding out that, you know, the oyster, the Pacific oyster, because its genome has been published, is one of the most studied organisms in this regard. We're finding out that invertebrates, everything from bees to oysters, have different levels of methylation. It turns out we're really different from mammals, and there's even some suggestion that maybe that repression activation thing might be inverted in some cases. But right now, some of the most interesting work is going on in oysters and in honeybees. So if you Google around and look for those things, that's pretty exciting. And the match that seems to be the most powerful right now is, for the future, is looking at the transcriptome and the methylome, all the epigenetic marks within your genome together. So in human biology and other model systems, people are trying to match those things to figure out what the regulatory mechanism is, how changeable and responsive that is. So it's like wading into a big gooey ball of complexity, thinking about mechanism where no one really has all the answers, but we do know that this kind of molecular biology does occur in cells, and we're wondering about its relevance in the environment. So one of the ways to break this down and think about it is really we're talking about genome by environment interactions, if you will, or organism-environment interactions. And there are really kind of two paths that we can go down. when I start talking about my study system, the purple sea urchin and their larval and embryonic forms, you can just think about out in the plankton, the larvae on the top one, and you can just ask something about developmental plasticity. We know that echinoderms are incredibly plastic. They're held up as one of the examples, the best examples we have of phenotypic plasticity. I don't know if you know this, but if you raise a culture of urchin embryos, when they get to that larval form after about 4 or 5 days, if you feed them a lot, they're in a food-rich environment, their little feeding arms are pretty short because they can get so much food. But if you starve them, their feeding arms grow freaky-deaky long, like they get just insanely long, trying to increase the surface area over which they can gather food. And that's the gene regulatory pathways that control calcification and the length of those arms reacting to the environment. So one of the best examples that we have of how this kind of interaction works. And so if we're just looking at developmental plasticity, we can query tons of things about the embryo, the larvae, how they respond to their environment with many, many different tools. But the one that we're kind of interested in from an environmental aspect is differential maternal investment and or exposure, because we know from a lot of work that what the female experiences changes the quality of the progeny, and we're now wondering what kind of mechanisms are working there and how does that play out in a global change context. So the study system I'm going to show you today from the one experiment is the purple sea urchin, Strongylocentrotus purpuratus. Eggs, gastrula, like an early forearm prism with the circles, the tummy in there, and those pointy things, it looks like a little spaceship, are the feeding arms starting to grow. And you can see these little rods, that's the calcium carbonate skeleton. The larvae is kind of like a backpacking freestanding tent where the calcium rods are the tent poles of its skeleton, and then the tissue is the cells or the tent itself over the arms. And in the plankton, different species stay out there for a different amount of time, but it's usually weeks while they're feeding, and then they'll settle out and metamorphose into a teeny tiny urchin and grow into a reproductive adult in 3 to 4 years. And just as a sidebar, people always ask me, well, are you going to grow multiple generations of these things? And I'm like, yeah, no. It's really hard to do. We can try, but there's a reason that there are not sea urchin farms and there are oyster farms, is because these are really hard to grow for a profit. And so that's why the sea urchin diving, the acquisition of uni, it's a diving wild fishery, so there are no farms for these guys. So you know, I'm going to tell you a little bit about what we're learning from this interaction and the urchins, but I want to do a little sidebar and talk about the environment a little bit. So these guys are native to the west coast of the U.S. We call it Bamfield to Baja. They're intertidal to shallow subtidal, but a lot of them occur in association with kelp forests. And this became a really important association for us because in about 2010, while we were wondering what the carbonate chemistry variation was in coastal oceans, we were able to collaborate with this guy named Todd Meritz at Scripps, who built a continuously recording pH instrument called the CFET. So we were able to get these out into the kelp forests around us and in other places, and it gave us the ability to look at, it's not perfect, you only get pH, you don't get DIC, etc., but you get a really nice continuous trace of pH that you can then use to calculate PCO2 in the water. And what we started to realize was that not everything looked like the open ocean, which is pretty smooth, because that's the kind of data we had going up to about 2010, coming from buoys like HOTS and BATS. But what we saw, two observations that are important. Once we had sensors out here in 2 places, Santa Cruz Island and Anacapa Island, and we have some on the shore too, but we saw this really distinct sawtooth pattern. And this, of course, is the photosynthetic activity of all the macrophytes, the kelp or seagrass. And in fact, that elevated purple line, we looked at that and went like, why is that different? And then we realized that there was a seagrass bed very close to that sensor that was just cranking in terms of sucking CO2 out of the water. So CO2 goes down, pH alkalinizes. So an oyster grower would call it sweet water. So the water gets sweeter when photosynthesis is running in the afternoon. And then sun goes down and the pH starts to drop because the plants switch into respiration mode and start releasing CO2. So this would seem to be not very significant, except that we are pretty clear now that this signature pattern really does influence performance of organisms that are in or near these beds of macrophytes. And many people right now are talking about how seagrass beds and kelp forests, not only are they blue carbon that sequester CO2 out of the water, but they're also places that will be probably refuges from future ocean acidification because of this pattern. And we have a lot of evidence that shows, even though there's flow through the kelp or through the eelgrass bed, the water still has long enough resident time to matter to the biology in there. So in a lot of ways, it's all of a sudden you go, oh gosh, it's not surprising that spiny lobster are recruiting to seagrass beds and loving it there, that there's some component of that we never saw before. So there are recent papers that have come out that literally are titled Seagrass Beds as Refuges from Future Ocean Acidification. And from a policy standpoint and a management standpoint, a lot of people are kind of interested in this because it's like, geez, restoration is good just to restore your ecosystem, but you also might be investing in future bucks in your climate change mitigation account because you're protecting this one area from future change. So there's also a lot of interest within agencies to think about that. So the other thing that's going on, the second thing, if we have macrophytes doing their thing, the second thing that happens on our coast is coastal upwelling. And most of you are probably familiar with this, but that wind comes from the north and basically it just drives surface waters offshore and kind of almost sucks up, we call it upwelling, that deep, cold and acidic water from the deep. And we know from research by Richard Feely and others that water shoals right up onto the coast, right up into the intertidal. So we can pick up signatures of upwelling in tide pools and in seagrass beds right on the coast with some of the sensors we've deployed. But this is a trace that comes from one of our Santa Barbara coastal LTR sites where you can see that upwelling happens, things are going along, it's all variable, and then boom, you have this big drop in pH because here comes the cold and acidic water. Those events last for days, usually 2 to 3 days. And it also comes with a very strong pulse of hypoxia. And low pH, upwelling, cold water, and low oxygen are highly correlated on our coast. So it is also a strong area of research, wondering what the consequences for that kind of signature might be in nature. Okay, so that's kind of the background. So finally, the experiment that I want to tell you about. So we called it Silver Fox because, like oceanographers, we didn't want to be left out and we name all our experiments something ridiculous. I don't even know what it stands for anymore, but we talk about it as it's something like, pH and oxygen, et cetera, changing. But the Silver Fox experiment, the idea was that we wanted to get adults and bring them into the lab before they were doing gametogenesis and hold them in the laboratory under simulated upwelling and relaxed conditions so that they invested in eggs as if they were totally happy, fine, pH is 8, and oh my gosh, I'm living in a really strong upwelling environment, or possibly a future ocean where pH is always 7.8. So we conditioned the adults, and then what we did after 4 months of conditioning the adults, we spawned them and raised cultures, but we did it reciprocally so that we switched the cultures so that progeny from each of the conditioned females were raised in the same conditions or different conditions as their mom. And then we measured a whole bunch of response variables, morphometrics, some biochemistry to look at maternal provisioning. We're just now analyzing our transcriptomic data, and we did a tiny little bit of DNA methylation study. So today I'm going to show you everything but the biochemistry, just little snippets of it to kind of frame out the story. So this is kind of the layout of the experiment where adults are conditioned for 4 to 5 months. We spawn and we get embryos, and we switch them into replicate buckets and raise them up. And and in here, we're going to call it blue and red, as you'll see. Relaxed is 500 microatmospheres of PCO2 at 17, and red is 1200 at 14. And those from our sensor data are like the 90 percentile, condition of upwelling versus relaxation. So regardless of, they're pretty ecologically relevant, but there's really good separation in what the adults are raised under. And we fed them kelp, once a week, and they were super fat and happy. They were like the best fed urchins ever. So we tried to keep them nutritionally equivalent during those 4 months. And then when it was time to go, we, inject them with KCl and collect eggs and sperm dry on the right. We do fertilizations just in one big old beaker and monitor that fertilization event until we have 95% success. And this is what the tanks look like when we have the adults in them. And then we switch over to pickle buckets real quick once we start to raise the larvae, so that the larvae are raised with the best high quality culturing practices, flow through systems, slow drip with stirring, and we have really high success raising them, low mortality, and we can also we're not macro culturing, but we can scale things up so we can put like 100 to 150,000 larvae per bucket and get a lot of individuals for RNA and DNA extraction. It's one thing if you're going to do morphometrics, you only need like 100 guys, but if you want to get enough RNA to do a molecular biology step, you need about 5,000 per step. So we had to learn how to scale our culturing practices. And then, you've probably all seen this from developmental biology, but, we then do a developmental timeline. So these are the experiments that take about 5 days to go from embryo to early pluteus larvae, and development waits for no one. So you're sampling gastral at 2 in the morning, and then you get 2 hours of sleep and you're up doing the next thing. So it's a really fun kind of community project. And the way we break these things down in the data I'm going to show you is we essentially have 4 groups of embryos, and we color code them with a 2-letter system. And let's see, the way I do it is the first letter is the mom and what condition she was raised in. So blue, just think nice, happy ocean, regular blue. And so a BB, the second letter, refers to what the embryos were raised in. So BB is blue mom with progeny raised in blue. And so you have 4 classes of BB, BR, RB, and RR, right? And our real question here was, were the blue moms different from the red moms in some way, in that their progeny had different performance because they did gametogenesis under really different environmental conditions? And so walking through the data, we're going to stick to this code, sorry. I usually use presenter view so I can cheat while I see the next slide ahead, and it's not set up like that, so I'm like, oh, I hope that's the next slide. So I'm going to show you 3 sets of data. The first one is the morphometrics. So my grad student Juliette Wong, who's leading these experiments, made these really cool slides for me so that the boxes show the color of the water and the insides are little representations of the stage. So those are tiny gastrula, and they're color coded to what they were raised in. So there's the BB, BR, RB, and RR. Okay, so let's first look at the eggs. So this was a kind of a classic response where the eggs were larger in the females that were conditioned, if you will, in the more stressful conditions. Everybody's fed the same. But this is kind of a classic result where the eggs from the red moms appear to be larger, and therefore we know, we're starting to get the biochemistry now, they have more goodies in them, if you will. So this is a classical invertebrate zoology outcome where a better provision egg usually leads to a bigger next stage or something, which in and of itself could be better fitness. You're bigger, you feed more, you become a bigger adult. And so if we then look at the next data set, so if red moms make bigger eggs before fertilization, we measured them. If you then run the cultures in that reciprocal experiment, you see that some of that size stuff holds, so that red moms, whether their progeny are raised in blue water or red water, they're always bigger than the blue babies from blue moms. So that too is not hugely surprising. from a larval ecology standpoint, because we kind of expect to see that. But we were very interested to see that the environmental signal that we were asking about, variable PCO2, high versus low, played out in this way. Because some people, when we did the experiment, would say, well, isn't that more stressful? You know, isn't that really like gonna hurt them, like deleterious? Like, shouldn't we expect that the red females somehow struggle during ametogenesis? And what we're learning is that tough mommies make tough babies, and there are ways that she's doing this that are quite interesting. And we're just now starting to see it. So if we go to the second thing, and this is just the snippet, it's kind of hard to look at because it's a lot of stuff all on one slide, but I wanted to do it this way. This is that kit that looks at total DNA methylation. It's not showing where in the genome the epigenetic mark occurs. It's just asking, like, how much methylation is there. So it's a little dodgy, but we wanted to do it because if there was a really strong, like, massive methylation signal, we would detect it. And there are other ways you can go in and look regionally for these things. So let's just walk through this. So if we looked, the first panel up there is just across stages. So interestingly, the eggs, and we've done this multiple times now, the eggs have really low methylation signals in the DNA that's there. So we're extracting genomic DNA and asking what's going on. So it's very low in the egg. And by the time you get to the early stage of gastroimpluteus, boom! So the methylation signal is coming on strong almost immediately after fertilization. And this isn't surprising either because purple urchins have been a developmental study model for decades. And what happens is there's a thing called the maternal zygotic transition, the MZT, and in different species that happens at different times where you go from using maternal RNAs to the embryo's own genome. And in purples it happens pretty early, like about, some people say as early as 2 to 4 hours they're starting to use embryonic genes, but by 15 to 30, like by the time an embryo is a gastrula, they're using their whole genome on their own. It's them. And so we see a really big upregulation of methylation if you just lump everybody together and look at stage. But in the next panel, in eggs, we had a lot of variability in our red eggs, but we were very suspicious in this experiment that there might have been some differences in that egg that was related to methylation. But when you start looking at gastrula, and it kind of fades out in pluteus, but gastrula is where that action is. It looks like red moms, whether babies are raised in blue or in red, they seem to have a little bit of a different methylation signature. This is really kind of data that you're kind of squidgy about because it doesn't tell you a lot mechanistically. But the information we're getting from this right now is that eggs are very different from early stages. We think maybe the eggs are a little bit different in methylation. And the next thing that we need to do for this is to use some other techniques that allow you to look at where methylation is occurring and to do full methylome sequencing. So that's kind of coming. But I wanted to show you this because these are the kinds of really simple assays you can do now just by getting on the phone and ordering a kit, which makes me sound lazy, but it's really nice to have someone provide that capacity to you. So this is the last piece of data I was going to show you. So in many ways, molecular ecologists, whether you're working on plants, who often lead, botanists often lead the way in molecular ecology, or animals, the transcriptome is a trait. And you can go into the literature and pull up a lot of papers that give you the developmental transcriptome for a particular organism. And this one is for a Mediterranean sea urchin. And what people do is just raise those cultures and then start looking at gene expression patterns across development. And so what we're trying to do is to look and see whether the transcriptome changes as a function of development and as a function of how the female was conditioned. And so we're just really looking at differential gene expression using a lemma. And we're looking for differences between the progeny from the red conditioned moms and the blue conditioned moms, depending upon what the progeny were raised in. And there is so much data here that just makes your head explode. Like the analysis of this is kind of amazing. But one of the emergent things that's happening is that progeny from red conditions moms, no matter what kind of water they're raised in, they are upregulating their transcriptome strongly. So they have a really strong transcriptomic response, meaning that more parts of their genome are activated at all times, no matter what they're being raised at. And so everything above 0 in this line means upregulation and differentially upregulated. So all those red dots are about 60% to 30% or so shifted below it. So there's this huge, almost like priming of the transcriptome in babies from the red condition moms. And so by looking at the whole data set, what we're really seeing is that parental conditioning really does matter. And we're starting to do some more performance assessments of the embryos and larvae to see how that performance comes out. Are you more thermally tolerant? Are you better at calcification? What is it about the change in your transcriptome that supports function? But the observations we have right now are really pointing to looking at gene classes and doing some further network analysis. But a couple of the snippets we're getting right now is that if you look at the gene classes, there's greater expression in the red embryo transcriptome for ATP production and ion pumping, which you might predict if you were facing dire low pH conditions. One of the interesting things we found that we did not expect was that we saw a downregulation of genes that are involved in epigenetic processes, so like those methyltransferases that lay down methylation marks. And we kind of were scratching our head, our DNA methylation data was really like, oh God, what does that mean? And reading in the literature, there's a lot of conversation led by people working on oysters and other invertebrates that limiting your DNA methylation might increase phenotypic plasticity using your transcriptome. So if you think about it, if you methylate something, you turn it off and you can't use it. And so the idea is that if you're operating in a really variable, crazy, environmentally variable environment, keeping all your transcriptic options open is ideal. So that some people are arguing that if you see low levels of methylation marks, that leads more of the reading frame of your genome open to respond to the environment. It's kind of a really tasty observation that we have right now, and we're still working through the data, but there are ways we can probably test that further. But that's kind of an interesting observation that's emerging from the studies that are coming out right now. And Steve Roberts up at the University of Washington is probably leading these observations, and I think he wrote a paper in Frontiers in Physiology to describe some of this stuff, so it's kind of interesting. So just by way of conclusion then, we really did see that conditioning of the female in these different environmentally relevant and maybe future ocean conditions really had a strong effect on the expression of the progeny, no matter what water they were raised in. We've got pretty strong evidence that transgenerational effects are in place, and that, in some ways, the female is somehow priming her progeny for experiencing an environment as embryos and larvae that might be as challenging as what she was raised in. And this is not terribly surprising because fisheries biologists have known this forever. Plant biologists know this, but we've never really kind of messed around with it in sort of an ocean global change context. Let's see, oops. The other reason I want to call this out in my silly little diagram is that out in nature, and we really want to understand what's going on in situ. This is my cartoon of a kelp forest, and here we have urchins that are in the forest. We also have urchin barrens way away from all the macrophytes, and we know that there's a really distinct environmental signal, very, very different across where those females are. And so we're kind of exploring this on a spatial scale as well, because if you were a manager, whether you're working in fish or corals, or you cared about the sea urchin fishery, if you knew there was a way that you could condition your adults or position your adults or do something to improve the quality of the progeny or the number and quality of progeny you get, you might want to do that. And so this has really practical implications as well. And we're really just right at the cusp of starting to explore the methylation signal and whether that's relevant in our system. So thanks for listening, and I'd be happy to answer any questions.
Is it tougher moms have tougher babies? Does it actually help them? And what about the survival of the progeny?
So did you guys hear that? What about the survival of the progeny? Are they tougher? They're bigger, so and have more fuel in them. Right now we know, we have like the first soupçon of evidence that they are tougher. We've done our first round of just thermotolerance trials, and it looks like all progeny from red moms have a better thermotolerance. So I don't know exactly what that means, but if I had to venture a guess, if your transcriptome is more available to you, there are a lot of genes in there that are part of the defensome, like heat shock proteins and other things to protect you and your membranes and proteins. And so if that part of the transcriptome is more available to you, it would confer greater thermotolerance to you. And there's been a lot of work in Drosophila that's kind of demonstrated that indirectly. So I think we have that, and we just did that last week. So I was like, no, I'm not gonna show that. But there are other things you could do that would support things like simple calcification assays, so like calcium labeling or just measuring calcification rates. Even with isotopes, like sometimes people will put these things in calcium-45 and do calcium labeling assays. So the other kind of performance that you would get from this might be feeding. That would be really interesting to do. And also swimming characteristics of these larvae would be very important. And all these things are like things I will never do, but I will call somebody up and say, hey, do you want some larvae? Because there's some really neat things here. Or even the structure of the skeleton. So the thing that we'd really love to do is to have an F2, for example, but it's so hard to do in urchins. I'm just accepting my fate and doing the molecular ecology. I don't know if I'll ever try to raise them or not. But you could choose a different system to do this in, like copepods, or like something that nobody wants to eat, but like Nucella, for example. There are some gastropod marine whelks that, breed really well in the lab, and you could raise multiple generations of them pretty easily. You can all think of animals that might do that. Paul?
So now that you have these transcriptome profiles of larvae that come from adults raised in different environments, can you go out to wild populations and look at those urchins that are in the seagrass areas that are buffered versus kelp forest and see those same signatures to show that that is what's going on in the wild?
Yeah, so we're starting that right now, and it's kind of like it's going to get done both ways. So we'll go out and we'll just do what we usually do, take in the adults and look at their in natura transcriptome, from the adults. But you could also do outplants. So take a bunch of urchins in from one location, do a common garden for a little while, smooth them out, and then put them out. That might be a better way to do, and then pull them out every now and then. The answer will probably be that they're quite different. I kind of want to do something that would be, I don't know, like use a mussel or a tiny mussel that is like a strong calcifier that could be like a tiny biorecorder. Just put them out in cages and measure their calcification and look at their transcriptome, maybe switch over to a different organism. But then you get a lot of pushback about, well, mussels don't really matter in the kelp, and so that's why you switch to surf grass and work in surf grass, which is dangerous. Yes?
So this is kind of following up
Yes.
on Noah's question a little bit, which is, so what it sounds like is that if you want to be a smart urchin, you just have everything fired up all the time. There's no costs to that. So there either have got to be costs, or why doesn't everybody just do that? You know, you've got all this variation.
Yeah.
Why not just hedge your bets and don't methylate anything?
Yeah, don't know, but great question. And the only place that I know that that's been addressed in some ways has been with the defense home and genes involved in stress, so things in Drosophila. If too many copies or overexpression of some of the chaperones occurs, it kind of does come at a cost because there's the maintenance of those proteins and then they sort of overfold things. But it's a great question, and I don't know if I really know what the selective pressure is. Why not have your every exon available all the time? Certainly there's a regulatory component in there that one would have to think about.
But your red versus blues ought to give you some insights into that.
So we're drilling into that right now, and it looks like, well, I mean, I guess you might be able to build some argument that building more mitochondria than you need is expensive. So we're going to start looking at what gene lists we have and try to see if we can identify pathways with some different analytical tools. But it's a great question, and I think it's kind of one of those things where you go down the rabbit hole and then you're like doing natural observations of the genome and you think, oh, I don't really know if this is all causal or not.
I can't really hear you very well.
Oh, sorry.
The acoustics in this room, just for
Oh, darn it.
For the powers that be in my department. I've been
Powers?
complaining about this for several years already.
All right.
This is a terrible room for seminars.
Okay, I got to talk louder then.
Because the acoustics are appalling. I mean, it's almost impossible to hear.
Okay. Do you have a question that I will answer very loudly? Yes.
The question is, to what extent do you think that, I know why you picked purple sea urchins. I mean, everybody studies purple sea urchins because Eric Davidson, made them the sort of the guinea pig for invertebrates, and their properties do seem to be very general in many respects. But do you really think that, in this kind of a respect, that these guys are also representative of what the mollusks are doing and what other echinoderms are doing and what the nereids are doing and the other phyla that are also present? Is this a general reaction?
I think the whole pattern is probably a general reaction, but I think if you think about physiological plasticity, like the way Massimo Pigliucci would back in the 80s, like it's very meta, like the ability to be plastic is itself a trait, and some of that plasticity is driven by the evolutionary history of the organism. So we also work in the Antarctic where the environment's really stable, cold, not this big sawtooth change in chemistry. And we see a very different response in a mollusk that we're working down there on pteropods. And so I feel like the emergent thing we're seeing is that organisms that live in variable environments kind of have that gestalt of greater, Uri everything, if you will. Of course, I drank the George Somero Kool-Aid and licked out the bowl, so that's one way to think about it. So I don't think they're representative of everything, but I think they're showing us a thing that's really important to me, and that is that not all species are created equal, not all oysters, not all urchins, and that therefore things will respond differently to future environmental signatures, changes in pH or chemistry. And so where a lot of people are working right now is the intersection of what's vulnerable and important via likelihood to have the environment change. So in some ways, that's why a lot of people are working on oysters, even though one of the oysters is an introduced species to the West Coast, but we love to eat it and we make a lot of money with it. So it's an important thing to look at in a climate change context. But thanks for that.
Yeah, really enjoyed your talk. Thank you. I had a few questions. One was about the trade-offs. I was wondering if you looked at the reproductive output of the organisms, and maybe that was the trade-off for gamete size, and then also just thoughts moving forward whether maybe another trade-off could be their ability to fight pathogens or deal, and this kind of goes into the third question is whether it would be worth looking at not just putting them into switching the type of environment, but putting them in a fluctuating environment and seeing how well they deal with that.
So going backwards, we did static conditioning and static cultures because we just wanted to start there. And we do have the capacity to change conditions that mimic the environment, so that's next. The second thing is, I'll do trade-offs. So a guy named Dan Okamoto did a lot of looking at investment in eggs and how gravid females were, depending upon how much food they had or how close they were to kelp forests. And there's definitely a signature there. We didn't have enough females to sacrifice them to do gonadal index, but that's something that we should and will explore once we can scale up our funky little setup to get more urchins in there. And the disease ecology question is really, really good. I think that's sort of an untapped phenomena here, you know, that there may be some trade-offs. There's evidence in other systems that preparing for environmental stress compromises you in the immune department. And we are kind of going to try to look at some of the genes that are related to the immune response. But again, the transcriptome doesn't really tell you everything, but it's a great point. And I think it's a really important area in global change biology right now, looking at the intersection of disease and a changing physical environment. Right on. Thank you for entertaining me the first time I told this story. So.